traits are being passed on, variation, those differences among those traits, okay, on the appearance of those traits that are being passed on from one generation to the other. We mentioned this a couple of times about asexual reproduction. Right? Meiosis, asexual reproduction, that is just like the focus of it. It is being genetically identical. Right? So asexual reproduction, single individuals or single cell organism passes those genes to the offspring without the fusion of any gametes. So there is no gametes such as like the sperms or the egg cells, or meiosis takes place in order for asexual reproduction. In order for asexual reproduction to take place. Right? So asexual reproduction, just basic mitosis. Okay? A clone, that's basically the group of genetically identical individuals from the same parent. So that's basically what it is. Right? Genetically identical, a clone. Asexual reproduction, mitosis. Here's some examples: the hydra and redwoods. Redwood can basically do it both asexual and sexual. And the hydra, this aquatic animal, the hydra, and that's where you have like actually the ecology. You've got one head, another head will come up, right? The regenerative capabilities of this organism is just like remarkable, striking. Here you can see like being budding out, right? It buds out, genetically identical, parent offspring, good, right? But if you were to cut this hydra in half, this part will grow left, this part will grow ahead. If you were to put this hydra in a blending machine, you blend it out, it will basically regenerate itself again. They are practically immortal, unless there is nothing to it, or unless someone else comes and eats it, right? So the amazing capabilities of how they're able to regenerate, it's just like mind-boggling. But that's what you see there, right? The asexual reproduction. Sexual reproduction, that's when two parents they give rise to offspring that they have unique combinations of genes inherited from the two parents. So you have the genome from the dad, the genome from the mom, you combine it, and then you have a new, unique genome in the offspring. So the offspring will be unique. The offspring will have its own genome that is not the same as the dad, it's not the same as the mom, and if they have siblings, it will not be the same as from the mom. So that we have the sexual reproduction. You can see here, right, parents and offspring, children, that they have their passing their traits, you can see similarities, and so on. So, whenever you see this, whenever you hear this, that you have your mother's nose, that is not exactly accurate. The traits are not being passed on. The genes are the ones that are being passed on, and those are the ones that once they are expressed, then those will be the phenotype, the appearance. Right? So, in a little sense, children, the offspring, do not inherit those particular physical traits from the parents. The genes are the ones that are being inherited. So somebody says this, you have your mother's nose, not accurate. Okay? You are passing on the traits, the genes, and those genes, once genotype ones become expressed, then they are the phenotype, the physical appearance. So the genes, those are those units of heredity, those are like segments of DNA that they are being passed on from one generation to the others, and they're being passed on via these vehicles, the gametes, right? These specters, so these vehicles, those are the gametes. Those gametes are how these genes are being passed on from one generation to the other. Sperm or egg cells. Sperm, you have male, females, egg cells. Right? So those are the genes, those are the gametes. And for these genes, they have specific locations, locus, that's where they have these segments of DNA, these genes in there. Right? So each gene has a particular location, locus on these chromosomes. And most DNA, that is the one that is packaged into the chromosomes. So this is the life cycle from one generation to generation, those sequence of stages in the reproductive history of an organism. So you have here the mature adults, then through meiosis they will produce these gamete cells, sperm cells, egg cells, and once they fertilize, they are going to have here this fertilization, a new organism, and that through mitosis is going to grow and develop into a full mature organism as well. Again, and then the whole cycle begins and again and again and again. So that is the life cycle. Compared to the lifespan is just basically the average number that these individuals they're going to be able to live, right? 70, 80 years old. And for sexual reproduction, opposite sexes produce progeny. So you need male and female as these gametes, and these egg cells, these sperm cells fertilize, and then you have the new individual. What determines the sex? The sex chromosomes. Right? We're going to see that more in a minute. Right, so for human somatic cells, those are cells in the body, your liver cells, your skin cells, all the cells in your body, somatic cells. They have these 23 pairs of chromosomes. So 46 chromosomes total, 23 pairs. 23 pairs because one pair comes from your mom, the other set of pairs will come from your dad. Right? So you have two sets. So you have 23 pairs. And in karyotype, that is the order display of these pairs of chromosomes from a cell. I will show you a picture of this technique in karyotype. And the two chromosomes in each pair are called the homologous chromosomes or homologous. This pair, basically one is from the dad, one is from the mom. Right? So homologous pairs, so they are the same. Homologous basically means that they are the same. They are the same size, the same length, the same shape, and they carry the same number of genes, the same genes, right? The same alleles. There will be some variation in there. For example, let's put an example, right? It's just basic example. Right? For example, like hair color, right? If your mom is like blonde, then in that particular chromosome, it has the same shape, the same size, the same length, and it has that particular trait in there, hair color, right? The dad, same, homologous chromosome, same shape, same length, same size, everything, and it has that particular gene in there, or hair color. But now here, for your dad, since he has like black hair, then he will have like black hair. Those are alleles, okay? We'll touch more that in the next sessions, those like differences, okay, among those appearances on those traits. Like alternative versions of the genes. We'll cover that in like more in the next sessions. But just keep that in mind. Those homologous chromosomes, they are the same size, same shape, same length, and they carry the same genes, okay? There might be some differences in the alleles, but it's the same gene, okay? Same gene, keep that in mind. So here's basically the technique. That's what you're seeing right there, the technique, the karyotype. I want to pause over here. This will be on the exam, okay? This technique called karyotype, and this is what you're seeing right here. The chromosomes, here you have here the centromere on the chromosome, the sister chromatids, the metaphase chromosome. They are lined up over here. Okay. So what is it that you're able to see on this technique, karyotype, okay? So one, you're able to see here the number of chromosomes. Here you have one pair all the way to the 22nd pair. So 22 pairs and one pair for the sex chromosomes. So, because you are seeing here the pair of the chromosomes, then you know that this is a diploid cell. Okay, so you have the two sets, one from dad, one from mom. Two sets. Notice here, right? This is the homologous chromosomes. One chromosome from mom, one chromosome from dad. They're the same size, same shape, same length, carry the same genes. Notice here the same color, right? Those are the genes. Those are the same genes that it carries there. Those are the homologous chromosomes. So it tells you that this is a diploid cell. It has two sets, okay? Two sets. One coming from mom, one coming from dad, okay? So that is a diploid cell. So 22 pairs, and here the sex chromosomes. So 22 pairs in total. These 22 pairs of chromosomes, these are known as the autosome chromosomes. In other words, the chromosomes that they will express, different traits for the body, autosome, okay, for the body, whatever it needs to express. And this one is the sex chromosomes. This is what it will determine the sex of the individual, the sex chromosomes. XY, you are male, XX, you are female. Okay, so diploid, right? It has two sets, one from mom, one from dad. That's what is there. You see the pair, then you know that it's a diploid. If you only see one set, so instead of seeing two here, for chromosome 1, you only see one, then you know it's haploid, okay? So we only will have 22 chromosomes, not 22 pairs. And we only will have one sex chromosome, not a pair of sex chromosomes. So only one, either the X or X or Y, okay? So it will be like X profile in the sex chromosomes, if it is haploid, okay? If it is the X cell, you know that it will have an X chromosome. If it is a sperm, then it could be either the X or the Y. Okay, we'll cover that in a minute. Alright, so it tells you this karyotype that is diploid, or it could be haploid if you only see one set, not two sets. Right? So it tells you if it is diploid or haploid. It shows you the autosome chromosomes and the sex chromosome. Okay? So that's what it's showing you right there. Also, it tells you right here, if you zoom in, then you will see this. You will see the sister chromatids. If you see sister chromatids, this is one chromosome, the one that comes from your dad. This is another chromosome, the one that comes from your mom. What you see here, that sister chromatids. What does this tell you? Through what phase they have undergone? Through what phase have they passed? The S phase. So you know that these are ready to divide, right? We go from the G1 to the S phase, and now it's the sister chromatids, right? G2 mitosis. So these are the metaphase chromosomes, so they're ready to divide, right? If you don't see the sister chromatids and you will see the one chromosome there, one chromosome here, no sister chromatids, then you know that it has not undergone the S phase, DNA replication. Okay? So, amazing technique. This is what it can tell you right there. Okay? So even though you don't see them here, but you see here the sister chromatids. You can barely see them here. Oh, these two little dots right there, right? That tells you those are the sister chromatids. So what can it tell you this technique, karyotype? Tells you if they are diploid or haploid. It tells you the autosome chromosomes, the sex chromosomes, so it will tell you what is the sex of this organism. And it will tell you if they're ready to divide or not, if they have undergone the S phase or not. Okay? So, amazing technique. So, make sure you know this technique, the karyotype. This will be on the exam for sure. Okay? Any question now? No? What is the name of this? Karyotype, okay? Right here, this technique. Make sure you know it. Do not misspell it. Do not say karyotype. That is not the right answer. Make sure you spell it correctly. And make sure you tell me what is the use. What does this tell you? Okay? Good. Right. So now let's go a little more to what we just mentioned, right? So I'm going to pass through this because I already referred to them. We just like to keep grinding the same thing. So the sex chromosomes, right? Those are the ones that determine the sex of the individual. It could be either be the X chromosome or the Y chromosome. So you are like human female, then you have a homologous pair of the X chromosome, X and X. You have X and X, then you are the biological female. One X comes from your mom, and another X comes from your dad. Okay? Human males, they have one X and one Y chromosome. So you are a biological male, you have XY chromosome. Sex chromosome determines your sex. So the X chromosome comes from your mom, and the Y chromosome obviously comes from your dad. Okay, X and Y. The remaining 22 pairs of the chromosomes, those are known as the autosome chromosomes that we have here. These are the autosome chromosomes, these 22 pairs. Okay, and this one right here is the sex chromosome, pair of sex chromosomes. Okay. So this one, the X came from whom?
Mom.
From the mom, and this Y chromosome came from?
Dad.
The dad, right? The mom cannot pass this Y chromosome. Okay. Alright, so autosome chromosomes, sex chromosomes. Okay. Somatic cells, and you have the gametes, right? Reproductive cells. Somatic cells, all the cells in the body. The gametes, reproductive cells, those are the sperm or the egg cells. In the sperm or the egg cells, then you know that those are going to be haploid. These are diploid. So you know that this is what? A somatic or reproductive cell?
Somatic.
Somatic cell, right? Diploid. Okay? Two sets, one from mom, one from dad. If you only see one chromosome here, in each of these boxes, in each of these pairs, only one set, then you know that it is what? Haploid reproductive cell, right? Is that what you said? I couldn't hear. Kind of, yes, yes. That means that it's haploid cell, that is a reproductive cell, either the egg cell or the sperm cell. How do you know if it is the egg cell or the sperm cell? How do you know if it will be the sperm cell or the egg cell? If I were to show you one of these.
The chromosomes?
The chromosomes, only one set, and here, right, the sex chromosome. If it is X chromosome, then it is what? Hmm?
An egg.
Who knows, right?
Oh, depending on
Because-
the set, if it's XY versus XX?
No. If it is haploid, right?
Oh.
Only one set, only one chromosome. If it has Y chromosome, you know for sure that it's a sperm cell, right? Only one, right? Let's say you don't have this. If it only has Y, only one set, right? Erase one of these, only one set.
Mm.
So instead of having 46 chromosomes, only has 23. So here only has one sex chromosome. Could be either this or that, right? If it is the Y chromosome, then you know that is what?
A male.
Sperm, right? You know that is a sperm. There is no doubt in that. If it is the X chromosome right there, then
You
what
don't
is it?
know.
Either, right? Could be either, right? You don't know. Could be either the mom or the dad or the sperm, right? If it tells you this one can only be X, then you know for sure that it's going to be the X, right? Alright, good. So keep this in mind, alright? Diploid haploid. How will the haploid cell look like? So only one set, not the two sets, okay? The sex, how do you determine the sex? The sex chromosomes, the pair of the sex chromosome, XY, XX. Right? If it is a haploid cell, reproductive cell, right, how will it look like? If it is the X cell or the sperm cell, okay? Somatic cells, reproductive cells, how will they look like? Once you have the skeleton, the autosome chromosomes, the sex chromosomes, make sure you go to distinguish them, okay? Any questions? Spend more time in here because this will be on the exam. Make sure you know the name, make sure you know what tells you. How will the somatic cell, how will the haploid cell be? Okay, perfect. Alright, so we mentioned this already. So in each pair of homologous chromosomes includes like one chromosome from each parent, that's what I keep mentioning, one from dad, one from mom. And these 46 chromosomes in the human somatic cell, there are like two sets of 23, so 23 pairs, one from the mom, one from the dad. Diploid cell has the two sets of chromosomes that we have there in the box of the homologous chromosome 1, two of them, one from the mom, one from the dad. Okay. So for the human diploid, the diploid number is 46. 2n equals 46. Okay. So 2n is a given diploid. Diploid number of chromosomes 46, 23 pairs. In a cell in which DNA synthesis has occurred, each chromosome is replicated, that which saw there, the sister chromatids, and each replicated chromosome consists of two identical sister chromatids. Okay. So for the gamete of reproductive cells, those are haploid. For these gametes, either the egg cell or the sperm cell, those are going to be haploid. Okay, so only one N. For humans, the haploid number is 23, N equals 23. Diploid number, that would be 2N, 26. Okay, so make sure you know that haploid number, diploid number. Each set of the 23 chromosomes consists of 22 chromosomes that are autosomes and one chromosome, which is the sex chromosome. Okay. In an unfertilized egg, the ovum, the sex chromosome is always X chromosome. That's what we mentioned, right? In a sperm, the sex chromosome could be either the X or the Y. So who determines the sex of the offspring? Who's that?
The dad.
Yes, the dad, right? Could either pass the X chromosome, so it can be a new baby girl, or it can pass the Y chromosome, so it can be a new baby boy. He says no. Why not? Why did you oppose that?
Because that is usually one a boy, baby boy. So
What was that?
Because that's usually one baby boy.
I couldn't hear you. I'm sorry.
Sorry, I was just shaking my head because usually guys want a boy instead of a baby girl. So I was like, that sucks. I think it's a choose, but not really.
Yeah, no. This is not saying that you're going to choose.
I know,
right?
but they're, you know.
Yeah. This is not. This is saying like, what determines the sex on the video, right? It is the sperm, right? depends on which sperm that fertilizes the egg, because the egg will always be X chromosome. Okay? Alright, so let's not get into the war of the sexes, so let's go back to this class, right? Seems like I want a boy, I want a girl, okay, fine, you will get your family issues, right? Alright, so here's the life cycle. We saw this before, but now this is more professional picture than the one before. So here you have the organisms, multicellular diploid adults, right, full-grown adults. Then in their reproductive organs, testes and the ovaries, you have meiosis, and these meiosis, those are the ones that are going to produce the gametes, right, the egg cells and the sperms, which are going to be haploid. So meiosis will be reductional, so it goes from N to N, right, from diploid to haploid. That happens in meiosis. And that is important because by alternating this number of chromosomes, then you are able to keep the same number of chromosomes in the adults. So now you are going to, through meiosis, reduce the number of chromosomes, so now they are haploid, and now restore to the same number in fertilization. Okay? Now they're going to be diploid again, once they fuse these gametes. So N and N, I will have to N. Okay? So make sure you know those key rules of meiosis and fertilization. Meiosis being haploid, fertilization now joining those gametes and restoring the number of the chromosomes. Okay. So here, that's what you have in fertilization, the union of the gametes, the sperm and the egg. And once this happens, then the fertilized egg, that is called the zygote, and it has one set of chromosomes from each parent, so diploid. One from that, one from mom. The zygote will produce somatic cells by mitosis, and that develops into another. So you see that with the role of mitosis. Meiosis, the role for that will be to produce the haploid cells, the gametes. The role of mitosis in this case will be growth and development. Repair, tissue renewal whenever is needed, right? So that's the role of mitosis. So at sexual maturity, the ovaries and the testes will produce haploid gametes. And these gametes are the only types of the human cells produced by meiosis, right? So not mitosis. Remember, mitosis will be for growth and development and for tissue renewal and repair. But meiosis will be the one responsible for creating these gametes. Okay. These gametes will be having these only one set of chromosomes, so that will be reduction. And that's what I mentioned here. Fertilization and meiosis, they alternate in the sexual life cycles to maintain the chromosome number the same, right? It was reductional in meiosis, then it gets restored in fertilization. If that were not to be the case, notice what will happen, right? If you only have this sexual reproduction, where you keep adding this number of chromosomes, the number of chromosomes, that will just be like keep increasing like exponentially, right? But because they are like half in meiosis, restored in fertilization, that chromosome number stays the same, unless there are some errors, which we'll be discussing in the next module. All right, so now let's go into the key points, okay? This will be like key differences for the meiosis and mitosis. And this will be like one of the main points for the exam. So, like mitosis, meiosis preceded by the replication of chromosomes. So that will be the S phase, right? Interphase, G1, S phase, G2. And this will take place before these cells divide. So meiosis will take place in two sets of cell division, that is called meiosis 1 and meiosis 2. But the set of DNA replication will only happen once before the meiosis 1, not in between. Okay? So only once. And these two cell divisions result in 4 daughter cells rather than 2 daughter cells as in mitosis. So mitosis, the result N is 2 identical daughter cells. In meiosis will be 4 daughter cells that are different from one another and they are different from their parents. Okay? That is the main key information. So the daughter cell has only half as many chromosomes as the parent cell. That is true meiosis. So keep that in mind, right? So mitosis, 2 daughter cells, genetically identical, equational, same number of chromosomes. Meiosis, it will be 4 daughter cells, will be reductional, half the number of chromosomes, and it will not be identical. They will be different, okay? Different from one another and different from the parents. So keep that in mind. Alright, so after each chromosome duplicates, then two subdivision follows, meiosis 1 and meiosis 2. Meiosis 1, that is the one that is reductional, so that's where it goes to half the number of chromosomes. And that is because the homologous pair, they will pair up with one another and then they will separate, okay? And that will result in the haploid number. Whereas meiosis 2, that will be equational, similar as mitosis. Equational means that it keeps the same number of chromosomes from when it begins to when it ends. Right? So that is meiosis 2, equational. And in this case, the sister chromatids separate, as they should, right? As you will see in mitosis. I will keep saying this thing over and over and over again. Meiosis 2 is similar to mitosis. There are some differences, of course, and make sure you know them for the exam, but make sure you know this key point, that meiosis 2, it is similar, not the same, but similar as mitosis. The main difference will be that obviously this will be non-identical, whereas in mitosis it is identical, genetically identical. We will cover that more in a minute. So the result will be for haploid daughter cells with unreplicated chromosomes. That's the end result of meiosis. So here you have the interface, right? This is the pair of homologous chromosomes. In a different parent cell, you have like one chromosome from dad, one chromosome from mom, and then you go to the S phase where you have the DNA replication. So now you see here the sister chromatids, identical, right? These sister chromatids are identical, and these sister chromatids are identical. This is chromosome 1, this is chromosome 1. So these are like homologous chromosomes, okay? Homologous chromosomes, okay? So these are non-sister chromatids, okay? These two are sister chromatids identical. These are sister chromatids identical, but these are non-sister chromatids, okay? They belong to the same homologous chromosome, right? Chromosome 1 from dad, chromosome 1 from mom. Right? So they are identical in the sense that they carry the same gene, but it might be different alleles, right? Again, let's say for example that you have the gene for hair color. In your mom will be blonde, but in your dad will be black, right? So they are alleles. They are like different versions of a gene, alternative versions of a gene. Right? But they carry the same gene. In other words, that they code for the color of the hair, okay? Or they might be the same. If both of your parents have black hair, they will be black for both, right? So it could be the same, it could not be, right?
So it's like the same category.
The same category, yes. The same, for in this case, for this particular trait, for your hair color, right? But it could be some differences. It could be the same, it could be different, right? But it's the same category, yes. So that is the gene right there, right? So these are identical to one another because they went through the S phase, right? These are identical to one another because they went through the S phase. But these, they might be or they might not be. These are the non-sister chromatids, okay? So keep that in mind. Whenever I mention, I ask you, identify or draw sister chromatids and non-sister chromatids, make sure you are able to identify them, okay? If I show you a picture like this, what I show you, hey, here are these homologous chromosomes. They might have more chromosomes, you have to find the ones that are homologous. In other words, same size, same shape, everything, okay? You have to find this and you will tell me these are sister chromatids, and these are non-sister chromatids of the same homologous chromosome. Okay? Alright, so it went through the S phase of replication. So now it goes through the meiosis 1. And in this case, the homologous chromosomes separate. Okay? Remember last time, whenever you guys did the activity right here, that's mitosis. The chromosomes line up in single file. Okay? Basically one line, right? You have kineticals right here that bond to the microtubules. You have the mitotic spindles over here. They're going to grab it, put it in the middle, and then separate, right? That is the metaphase, right? Metaphase split. Chromosome line up in single file, one line. In meiosis, you will have the homologous chromosomes pairing with one another. Okay? Like that. Instead of being in one line with these 4 chromosomes, let's say this is smaller. These are big, small. They will pair up with one another. Okay? Let's say this is red. This is red. That means that this is from your mom, this is from your dad. From your mom, from your dad. They make one line, okay, and they will be separated, right, in mitosis. Here, that's the one from your mom, that's the one from your dad, okay. This one could be either or, right? I'm just for the example, I will put it right here, but we will put that in a minute, how they can arrange independent of sorting. Here, that's from your mom, that's from your dad, right? I want to point out here the main difference for meiosis and mitosis. Mitosis, single file, one line. Mitosis, double file, two lines, okay? Can you see this difference right here? Meiosis. This is the basis of separation of these homologous chromosomes that will create the haploid number. Because once you separate these, right, these sister chromatids, they become a chromosome, right? And now you have two identical daughter cells. In this case, when the cells divide, this cell will have two chromosomes, and this cell will have two chromosomes, right? It went from having 4 chromosomes to now the two daughter cells only having 2 chromosomes. Can you see this? Yes or no? Should I repeat it? Or you guys are fine with that? No? Again? Raise your hand if you want me to explain it. Okay, okay. I need 4 people over here. You two? Right here? Somebody use you. And 2 more. I think you raise your hand and you raise your hand. So come over here. Alright, so here we have 2 chromosomes, right here 1, right here 2. So you want to pair each other with one another, so these are the sister chromatids, right? Sister chromatid, 1 chromosome. Sister chromatid, 1 chromosome. Okay? Good? Alright, perfect. So let's do mitosis. Mitosis will come over here. So you will come over here, and then you guys will come over here, right here. Okay? That's mitosis, one line. And then let's say if the cell has many more chromosomes, they will line up over here, all the way, to the end. So in this case, only how many chromosomes?
2.
2 chromosomes, one over there, one over here, right? They have the sister chromatids, so that tells you that they have undergone to the S phase, right? Replicated, right? Because they want to divide, right? So then we go to the anaphase. The whole cohesion is there, is bound right here. Separates will come and it will cut them right here, right? So now you go over there, you go there, you go there. Right there, right there. So now here we have one cell, here we have another cell. How many chromosomes here and how many chromosomes on this cell?
4.
How many chromosomes are on this cell right here? 2, right? How many chromosomes on this cell? 2, right? They have separated, right?
Yeah.
Let's go back to the original position. This is the parent cell. How many chromosomes in this parent cell? 2 chromosomes, right? But they have the sister chromatids, but still 1 chromosome each, right? Now the cell divides. Let's go and divide. And now here, the line of demarcation, that's 1 cell. This is 1 cell. How many chromosomes on this cell?
2.
How many chromosomes on this cell? 2. So notice that by doing the single file, the number of chromosomes remains the same. Parent cell 2, daughter cells 2, right? Let's do meiosis. Go back to your sister chromatids. So now, instead of pairing up in one single file, now they're going to pair up in double file. You pair up next to pair, right there. There you go. Now they pair up like that. So how many chromosomes do I have right there?
2.
2, still 2, right? This is called a tetrad because now you have 4 sister chromatids, right? Sister chromatids are chromosome 1 from mom, sister chromatids are chromosome 1 from dad, right? So they're identical, sister chromatids. These are non-sister chromatids. They are not identical. They could be, they could not be, right? But they are not identical, okay? Non-sister chromatids. These are the tetrads, 4 chromatids right there, okay? So in meiosis, they're going to separate, but they're going to separate right here. Go over there, over there. So now here we have 1 cell and here we have another cell. How many chromosomes on this cell? 1. This is 1 chromosome, right? They have not been separated. How many chromosomes right here? 1. Only 1 chromosome right there. So notice how it went from 2 chromosomes, right? Whenever they were grabbed right here, so to come over here close by. This is the parent cell, 2 chromosomes, right? One here, one there. But once meiosis takes place, meiosis 1, the cell divides right there, it goes right there, it goes right there. And now here in this cell, how many chromosomes?
23?
No, on this example.
One.
On this example.
One.
On this
Oh,
example.
one.
1, right? And in this case, 1, right? If it were to be like human cell, then yes, it will go from the 46 to 23, right? But just to keep the example simple, it goes 1 chromosome here in this cell, 1 chromosome in this cell, okay? So now you have meiosis 1, 2 daughter cells, right? Which has been reduced, the chromosome number. It went from 2 to 1, or in human cells, from 46 to 23, right? Once this cell divides over here, then this cell divides right there. Sister chromatids separate, and now here you have 1 cell, here you have 1 cell. How many chromosomes in this cell?
One.
How many chromosomes in this cell? 1. So meiosis 2 is equational. It begins with 1 chromosome, the daughter cells are 1 chromosome. This cell will divide 2, and now how many chromosomes here? How many chromosomes here? And then you have meiosis 4 daughter cells with reductional number of chromosomes. Okay? Does that help?
Yeah.
Okay? Okay, good. So
We're
let's
just racking up extra credit points, huh?
We must
Yes,
have a
it is.
lot.
Alright, so that's what you have right there. This is the last dynamics that we just did. Right? So at the end, you have here meiosis 1, homologous chromosomes, they will separate, so this is reduction, so instead of having 2 chromosomes, now you only have 1, and then from these cells, it will separate, and then at the end, you will have 4 daughter cells with the reduced number of chromosomes. Okay? Any question on this one? Okay. All right, so meiosis 1 is preceded by the interphase, we mentioned this before, in order to duplicate the DNA, sister chromatids. Then these sister chromatids are genetically identical. They are joined at the centromere, as we have seen before. And the single centrosome replicates on 2 centrosomes. So same as we saw before, right? Cell division mitosis. And now here you have the first cell division, that is meiosis 1. It's composed of prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. That's what you have here, meiosis 1. Then after does this, then you have meiosis 2, right? And at the end, you will have these 4 daughter cells that are in haploid mode. Okay. So let's go a little more into about each one of these. So meiosis 1, that is, whenever you have this separation of the chromosomes, you have here prophase 1. Notice what happens in prophase, then eating, DNA condenses. But here in meiosis 1, you will also have here this chiasmata, okay? This is known as the synapses, whenever they will line up nucleus with one another. These non-sister chromatids of the homologous chromosomes, okay? They will pair with one another, they will form this chiasmata, that is the X shape, where the genetic exchange takes place. So they begin to exchange that genetic material, those genes, among those homologous chromosomes. One chromosome from the dad, homologous chromosome from the mom, they begin to exchange those alleles, that genetic information. That's what takes place right there, the chiasmata. And then for the metaphase 1, the homologous chromosomes, they pair up with one another. So 2 lines, don't fight, just what we did right here. Okay, so 2 lines instead of being just 1 line. 2 lines right there. And then anaphase 1, they separate. Homologous chromosomes separate. Okay. Not the sister chromatids, the homologous chromosomes. Notice here how they have these chromosomes with the ends of the sister chromatids different color. That's because of the genetic we have mentioned.
Well, why do they show it in metaphase then?
That's where it's taking, yes, they are showing it right there. Here's where it's taking place, and here's where you can see them. Maybe you don't see them because you need to zoom in, but here you can see this chromosome, how the end of the arm is red. Okay? Same thing over here, you can see that it's red. You are not able to see it really careful, or really like fine resolution right there, but you can see that it takes place right here. Here they line up, next to each other, and then here they're separated. Okay. homologous chromosomes separate. And then you have telophase 1 and cytokines, pretty much the same, uncondense and then divides. Okay. On prophase 1, this is what happens, right? This typically occurs more than 90% of the time for meiosis, and then chromosomes begin to condense. But this is one of the main distinctions of prophase 1, the synapses. That's where the homologous chromosomes, they loosely pair up, aligned gene by gene, close with one another. Once they do the synapses, then they form this chiasmata, right? The X shape, that because of the crossing over. Where these non-sister chromatids, they exchange these DNA segments. Think of, for example, here, whenever you have these two chromosomes, let's say one of them, sister chromatids of chromosome 1 from that, begins to exchange information with one of them, the non-sister chromatid. The chromosome 1 from that. From mom, from that, they begin to exchange information. So that's pretty much the crossing over. Exchanging these DNA segments, this DNA. And because they are the same homologous chromosome 1 from that, homologous chromosome 1 from mom, they carry the same gene, so they are pretty much like exchanging information among the same categories. Okay? So that's what is happening there. Exchange. On the metaphase 1, you see these tetrads, these 4 chromatids, right? Homologous chromosomes, one from that, one set of mom, one set of that. They line up at the metaphase place with one chromosome facing each pole, right? Yes.
Can you explain again what tetrads are?
Okay, come back over here. You too, over here. Yeah. So you complete this.
I'm sorry I said anything.
No, I'm pretty sure somebody else had the same question. So, right. So, let's do meiosis right here, right? Chromosome 1, sister chromatids right there. Chromosome 1, but one from mom, one from dad. Sister chromatids of homologous chromosome 1. So here you have chromosome 1 from mom, chromosome 1 from dad. Okay. Sister chromatids, so they're identical. Okay, they're identical one another. Here, sister chromatids, they're identical with one another. These are non-sister chromatids. Okay. Here, whenever they come together, right over here, they loosely pair up. That is synapses right there. Loosely pair up.
You don't need
right?
to touch?
Yeah. They loosely synapses. They loosely pair up, aligning next to each other. Okay. That we're seeing right here. Synapses. Okay.
Do we touch? Kinda touch?
Kind of touch, yes. But then, let's say, can you raise your hand?
Are you saying synapses?
Synapses.
Okay.
And then raise your hand. There you
Like
go.
that?
Yes, and then
Okay.
change. Okay. This
Oh,
is
okay.
genetic rearrangement. Okay.
Crossover?
Yes, crossover. Okay. Genetic rearrangement. The synapses, the X shape, the chiasmata, and the genetic rearrangement. Now think about that she has now this arm and she has now this arm. Genetic rearrangement. Okay. Chiasmata, X shape right there. The X shape that you see right there. Okay. All right. So that is the.
Tetrad.
That's the tetrad right here. Okay.
Oh, okay,
These
okay.
4 sister chromatids.
Oh, okay.
So
So when they're mixing.
they are mixing, yes, mixing. Tetrads, these tetrads. And now that, could you keep raising your hand? All right, so whenever they separate, this is metaphase, okay? They line up right here. They line up in the middle, these tetrads right here. These are, tetrads basically means for 4, these 4 sister chromatids right here.
You said facing each pole, right?
So that's right here, okay? For this pole, there is one pole, magnetic spindle right here. The macrotubules, they will bind to her kinetical right here. Macrotubules from this pole right here will bind to her kinetical right here, okay?
It's not visible.
It's not visible, yes, unless you want to Gaspar and everybody
Yeah,
else.
everybody knows.
Yeah, for real. We're up here, you guys gotta be up here too.
Oh, you guys get the points, all right? Extra points. All right, so
Wait,
whenever.
what?
She's recording.
All right, just for the sake of time.
Where's everybody at?
I don't know. Yeah, well, anyway, yes. So once separate, so you guys go over there, while the magnetic spindle here, begin to pull right there, and you can pull
This is the
right there.
anaphase?
This is the anaphase right here, right?
And this is when we have a mixture of her.
Yes,
Yeah.
I was going to take out her sweater and put it over here, and her sweater and put it over there, right? That is the genetic rearrangement. Just keep that in your mind, right? So that is the genetic rearrangement, okay? So that's the tetrads, that is the synapses, the chiasmata.
We are not gonna separate, right?
Hmm?
In anaphase, we don't separate?
Not right here, not in anaphase 1. You will separate in which one?
Anaphase.
Anaphase 2. Anaphase 2. Okay? Anaphase 2, you guys will separate. Okay? Alright, good question? No. So anaphase 1, what separates? Sister chromatids or homologous chromosomes? Homologous chromosomes separate in which one? Anaphase 1. That's right, correct. Good. Alright, so this is basically what I showed you, the microtubules from each pole, right? They will basically attach to the kinetochores, and that basically, what is leads to the anaphase. Anaphase 1 is pair of homologous chromosomes, that is the one that separate, then move towards each pole, that is spinneraphovirus like that. And then the sister chromatids, they will remain attached, they will not separate until the anaphase 2, right? So the sister chromatids remain attached, the centromere, and they move as one unit to one pole. Okay. Then you have telophase 1 and cytokinesis. That is, the uncondensed and still has the sister chromatids right there, and then the cytokinesis, division of cytoplasm. And then here, that's what produces the two daughter cells that are reduction of chromosomes, that haploid. It began with diploid, and now that haploid because the whole homologous chromosomes separate. Okay? Good? Right. So in animal cells, you have the cleavage spora, right, the cytokinesis. In plant cells, you have the new cell plate formation to the Golgi process. There is no chromosome replication between the meiosis 1 and meiosis 2, so there is no need for that. Okay. Then it goes on to the meiosis 2, and then also it has these four phases, prophase 2, metaphase 2, anaphase 2 to the phase 2 and cytokinesis. This is the one that I was mentioning. I already mentioned this like 5,000 times, right? Meiosis 2 is very similar to mitosis. Not the same, but very similar, right? In the sense that they separate in like similar fashion. The chromosomes that will line up in single file, sister chromatid separates at the metaphase, and then you will result in the two daughter cells at the end, right? Yes?
I thought you were saying meiosis 1 was similar to mitosis, no?
What was that?
I thought you were saying meiosis 1 was similar to mitosis on the board, no?
No, it was meiosis 2.
Oh,
the
meiosis
one that
2.
is similar to mitosis.
Okay.
Okay. So here you have meiosis 1. In meiosis 2, you have this separation right here, right? So at the end you will have these right there. And these right there. So now in meiosis 2, this cell will line up in single file, right? And these will separate. And now having here only these sister chromatids separate to create the haploid cells that you can have, right? With equation we know, right? Because now here you have two chromosomes, right? Here you have two chromosomes, here you have two chromosomes, here you have two chromosomes. So it's equation in the cell. So this meiosis 2 is similar to mitosis. Yes, question?
Could you use a different color? Because I can't.
I know, but I don't